we've done has been more scientific, but I'm really excited to be able to see more of a clinical presentation for some part of this conference. So, Richard, please take it away.
Does this work? Oh my God, it does. Thank you very much, Adam. I certainly hope we can live up to your hopes in the work that we do. So I guess it'll be switched over. So yes, I'm a clinician. I do run a diagnostic laboratory, so at times I can bootleg some research off of the chemical machines that we have in the laboratory. But I am essentially 100% clinical. But I use what I wanted to present today and emphasize is that even though biochemistry is old hat and I'm a fossil, you really can learn a lot from looking at a patient and just using ordinary biochemical methods available to any doctor, amino acids, organic acids. I first got interested in Barth syndrome because of methylglutaconic aciduria, which at that time, back 25 years ago, was a very unusual finding. Only 2 years ago did we figure out why the boys with Barth syndrome have methylglutaconic aciduria, but I hope progress in the future will be a little bit faster. So I'm going to review some findings that they really began to move ahead with some real gains in 2008. At the 2008 meeting, we obtained metabolic studies on a large number of boys, anywhere from 18 to 25, depending on what tests we were doing. And we were able to, in contrast to the kind of data we can collect that is collected in the registry, the timing was very precise. And for metabolic studies, that's very important. So by doing amino acids and fatty acids and some other parameters, we were able to get a real window on what's going on in the biochemistry, and that combined with a number of the molecular findings really came together, I think. And I hope I can convince you they've come together in an understanding of some of the basic pathophysiology of Barth syndrome. So let me... What? Is it on? And is there a switch?
Okay, it's not working. Oh, there.
There it goes. So what am I doing?
It's supposed to work.
Where is it shooting? Over there?
Here, it's on now.
Okay. All right. Oh, good. So I start off with a provocative question. Is Barth syndrome a mitochondrial disease? And obviously it is a mitochondrial disease, but the problem I feel in getting ahead on our understanding of Barth syndrome is that it's a very atypical mitochondrial disease. So the paradigms for mitochondrial disease are apoptosis and oxidative stress, deficiency of ATP, and all of that. And we found that just doesn't hold true in Barth syndrome. For many years I tried treating it with antioxidant cocktails. It has absolutely no effect. And it's also, in that I deal with a lot of mitochondrial disease, I can say it's very atypical. And that, as I list here, there are some paradoxes in Barth syndrome that require some explanation and which should guide our thinking in terms of how to determine where the primary problem is. So the muscle biopsies sometimes have had normal enzymology. Clearly some of them show abnormal mitochondria, but the enzymology can be completely normal. Compared to other mitochondrial diseases, the muscle weakness and fatigue is so severe despite really almost no typical biochemical signs of mitochondrial dysfunction. So that's really unusual. The severity of the growth delay also is completely out of proportion to the problems in mitochondrial disease. We have patients with like complex 1 deficiency where the growth in fact is is faster than normal many times. So growth delay, to see growth delay in a mitochondrial disease because of the mitochondrial disease, it's got to be really bad. High lactic acids, severe failure to thrive. But these boys just have classical constitutional growth delay. So it does not look like a metabolic growth failure. And the ATP synthesis has been demonstrated 10 years ago or so is basically normal in cultured cells. There are some deviations from normal in some of the parameters, but the cell makes normal ATP. Actually, I think I'm going to bypass this entirely. So the cardiac pathology in Barth syndrome, I'm sure everyone is familiar with it. It's a dilated cardiomyopathy. It can be stable or progressive, and it can vary quite a bit. That is, the child can go into heart failure over a relatively short period of time, a few months, and then just as quickly get out of heart failure. That's also, for unexplained reasons, it's not clear sometimes why they get better. There's increasing, oh dear. I'm a clinician. Okay. That can wait. And one of the other things that's puzzling is that the ventricular arrhythmias can occur even when the cardiac function is completely normal, which is, of course, in cardiomyopathy, the arrhythmias usually are in relation to the degree of cardiomyopathy. And the mitochondria are normal or abnormal, and in vitro ATP synthesis is normal. So many years ago, in the first family that I saw, at the time I was working with Britton Chance doing studies on NMR spectroscopy in muscle. So this is a system where one can measure ATP levels in vivo. So the NMR is gated to the heartbeat, and it measures the phosphorus molecules in the living tissue. And typically what is seen, I guess, I can't get the pointer to work.
There's an alternate one up there.
Oh, great.
The red one does work, but the green one's much brighter.
Okay. So this is an ATP signal. There are other ATP markers, but usually this one is free from contamination, and this is used as a marker of the level of ATP. And PCR is phosphocreatine, which can be viewed as a source of reserve ATP. And this is a pair of twins, one of whom has Sanger syndrome, which is a defect in complex 1 in most cases. And what it shows is that the ATP level is normal, and the reserve is low, but again, the ATP level is quite normal, and which is to say, and this is a theme that I'll come back to several times because I think it's very important understanding the treatment of Barth syndrome and the treatment of other mitochondrial problems. So this is the boy with Barth syndrome, 8 months old, out of heart failure, but still has a severe dilated cardiomyopathy, and if anything, the level of phosphocreatine, the ratio of phosphocreatine to beta ATP, which is considered a sign of energy reserve, if anything, it's on the high end of normal. It's definitely normal. So ATP synthesis in an abnormal Barth heart is completely normal. That's not what people think of in a mitochondrial disease. Even the ATP reserve is normal. And in studying other mitochondrial problems, there are some other patients that I have studied. This is the child with Sanger disease, with Sanger syndrome, who the PCR beta ATP is low. Again, the ATP is normal. Here's another child with lactic acidosis, a mitochondrial problem. And here are some sibling controls. And here's a patient with Barth syndrome. So again, very different from a typical mitochondrial disease. So we have to think very differently about what the pathophysiology is in Barth syndrome. This just shows the gastrocnemius, and again, this boy is extreme. Well, 8 months, he's not walking, but when he's walking, extremely fatigued, and can barely get around, and yet there's no deficit of ATP in his muscle. So I skipped past the slides that talk about cardiolipin. I really just wanted to show those to say that cardiolipin, to remind everyone, cardiolipin is a major component of the intermitochondrial membrane in which sits the electron transport chain. So when it was discovered that the primary defect in Barth syndrome leads to a deficiency of cardiolipin, of course, most people thought about, well, this is it must be affecting the electron transport chain. And like in any other mitochondrial disease, that makes perfect sense. But again, if it affects the electron transport chain, why is ATP synthesis normal? So that's a real puzzle. So now I'm going to turn to, so we have these puzzles. We have to explain why Barth syndrome is as severe as it is despite normal ATP synthesis. And otherwise, except for the heart failure and weakness and the other problems we'll come to later, neutropenia, can be healthy boys. So now I'm going to turn to what we do clinically. So we see a patient with a mitochondrial problem, a metabolic disease. One of the things we look at is amino acids. And we have made an effort to standardize very carefully how amino acids are measured, because they serve as a very important marker for what's going on inside the mitochondria. Most amino acids, essentially all amino acids, are metabolized through the mitochondria, and so serum amino acid levels their levels are regulated by mitochondrial function. One of the most common things we see is an increase in alanine here in mitochondrial disease. And what I'm showing here is that we always collect the blood at 4 to 6 hours fasting. That's what we did here in 2008. The reason we do it at that time, and specifically that time, is that's after the artifacts of the amino acids that come from a meal, the immediately digested protein, that's gone by 4 to 5 hours usually. And it's before there are secondary changes in the amino acids that are caused by fasting. So between 4 to 8 hours or so, the amino acid levels reflect mitochondrial function, many of them, and their ratios stay constant, more or less constant as they go down slowly, but they stay constant. So if you want to collect the amino acids in a controlled manner, not just at the end of an office visit, one can gain a lot more information from the studies. And I'm just showing here that it happens to be that the mean in our laboratory is very close to what we get at 4 to 6 hours fasting. And so one of the ways we analyze the amino acids is to express not the absolute level, but the ratio to the 4-hour mean. And so if someone has a mitochondrial problem, the alanine might be within the technically normal range. Some laboratories have a normal for alanine up to 600. But relative to all the other amino acids, it'll be high. So the alanine ratio to the normal mean at 4 hours could be 1.8, which is clearly pathological, even though it's in the normal range. So one can take a report from a laboratory that says no abnormalities, analyze it in that way, and make a diagnosis. And most of the time when I'm asked to see a patient with a mitochondrial disease, or to determine whether a child has a mitochondrial disease, I just send me the amino acids first. And more often than not, I call back and say, here's your diagnosis. So there's a tremendous amount of information that comes from the amino acids, and you'll see how this is important with regard to Barth syndrome. So this is what we found. Oh boy, this is what we found in 2008, looking at the amino acids. So here these are controls based on 4-hour fasting. So this is the range of normal for cysteine, methionine, phenylalanine. The controls in Barth syndrome are very similar. But look at arginine. There's no question that is low. And if one obtained the amino acids like 2 hours after a meal, the arginine would be completely normal. Now, and this actually is within the normal range of most laboratories. The normal is 15 the lower limit of normal. So many of these boys would have an arginine level that's in the lower limit of normal. And also, arginine is unstable, so if someone sees a amino acid profile in Barth syndrome and sees a low arginine, they say probably breakdown of arginine to ornithine. And indeed, ornithine, for other reasons, is increased in Barth syndrome. Is this significant? Well, yes. We measured prealbumin, which is a measure of the intrinsic ability to make protein. And anyone who's malnourished will have a low prealbumin level. And the prealbumin was clearly low compared to controls. And what's interesting, I don't have the plot here, but we also collected BNP, the natriuretic peptide that correlates to mitochondrial function. So if someone is in failure, the BNP is high. And there's almost a linear inverse correlation. As the BNP went up, the prealbumin went down. So it's to say that inability to make protein correlates with with the condition of the heart in Barth syndrome. So we're all right here getting a clue. This is nutritional, not metabolic in the classical sense. So that was an interesting finding, but how do we explain it? And this is what gave us the most important clue. So I follow a family, I've followed for many years, a family with autosomal recessive Barth syndrome. When I say Barth syndrome, it really is. It affects girls and boys. We have a family of two boys and one girl, and they have all the features of Barth syndrome, except the abnormality also affects the brain. And as you know, in Barth syndrome, the cardiolipin abnormality is not manifest in the brain. But these children have brain disease, and it causes progressive problems, including seizures, Parkinsonian tremor. They have developmental delay. And we early on excluded TAZ as the cause of this, I worked with a group in NIH to find the gene. And at the same time, another family of dilated cardiomyopathy in the Hutterites in Canada was being studied. And we both came up with the same diagnosis in the two families. I'm just showing here that the methylglutaconic acid, this is unaffected, the methylglutaconic acid is also increased. So all the features of Barth syndrome, biochemically and clinically, are present. They also have hypercholesterolemia, as you do in Barth syndrome, and they have low plasma arginine levels. Not quite as low as we see in Barth syndrome, but they do. And they also have a citric acid. They have a greater citric acid cycle organic aciduria than patients with Barth syndrome. So this is molecular people probably understand this. I know there's control, parents, heterozygous, and a deletion. And this is in DNA JC19, which is a major protein in the intermitochondrial, one of the intermitochondrial membrane protein transporters. So this is the TIM23 complex that sits in the intermitochondrial membrane, and DNA JC19 is equivalent to TIM14. So with that, it was immediately obvious, or it seemed the most likely hypothesis or explanation, is that the comment to have such a similar syndrome caused by an abnormality in cardiolipin and an abnormality in the function of the protein transporter, the argument is one of the most significant effects of the cardiolipin deficiency is on the function of the TIM transporter. I mean, that's obvious. So the question is, what exactly is that doing? Well, the TIM23 complex transports mostly as matrix enzymes. There's another complex for transporting the electron transport chain, and that's normal. The TIM23 complex is for matrix enzymes. And what's interesting is that TIM17, which is the other half of this complex, is the only TIM protein that comes in two forms, an A and a B. And if you look at the expression of the TIM17B, it's interesting how it parallels the abnormalities in Barth syndrome, that TIM17A is not expressed as much in muscle and heart, but the TIM17B. So one of the hypotheses that comes from that is that the cardiolipin deficiency is affects mostly the TIM17B. Now, there could be other explanations for it. It could be coincidence, but at least it guided our thinking in terms of what the underlying functional problem is in mitochondria. And our esteemed colleague, Dr. Greenberg, had shown in 2000 that indeed there is an abnormality in matrix enzyme import in a yeast mutant, a yeast cardiolipin mutant. And of course, I was very prescient that we, in 2000, didn't pick up on well enough. So here's the here's the citric acid cycle, and I apologize, but you're going to become quite exhausted looking at the number of times you have to look at this. But it really is so fundamental to our understanding of Barth syndrome and other mitochondrial problems. So if arginine is low, why is arginine low is the question. Well, when there's a block in the citric acid cycle, the mitochondria have methods of rescuing ATP synthesis. That is, if there's a decreased production of alpha-ketoglutarate because, let's hypothesize, there's a block at isocitrate dehydrogenase, then arginine is pulled in as a substrate, as an alternate substrate for alpha-ketoglutarate. And then the citric acid cycle can continue. It's been shown in a number of research studies that if you decrease flux if you significantly decrease flux in the citric acid cycle, the cell dies. It's as simple as that. So if the cell has an abnormality in the citric acid cycle that cannot be rescued by some method such as this, then the cell will die. Arginine, for some reason, seems to be picked on, at least in terms of what we measure in blood. We can't, of course, tell much about flux by looking at blood, but arginine is low. These are not low in particular. But so one would hypothesize there's a block here at isocitrate dehydrogenase, which is involved in both of these steps here, but it's basically involved in the conversion of isocitrate to alpha-ketoglutarate. So what we wanted to do next, of course, is measure levels of isocitrate and cisaconate. And I was surprised when I looked in the literature that there was no method, no clinical method for doing that. It's been done in research studies, but no one had looked at levels in plasma. So we set up the method, and this thing is jumping all over the place. And that's what we found, that isocitrate and aconitate were significantly increased over the other metabolites. So it doesn't necessarily prove that the block is in the import of isocitrate dehydrogenase, but it certainly is a working hypothesis that that's where the problem is, that the TIM complex is most impaired, and that what is most affected by the impairment in the TIM23 complex is the import of isocitrate dehydrogenase. Maybe aconitate, but the aconitate probably is just a backup or could be a backup from that. So again, isocitrate, cisaconate are increased. One of the other interesting things that come from this, if we say there's a block here, what happens to all the citrate? Now when you measure citrate, it's not high. It's a little bit high, perhaps, on average, but it's not significantly increased. And that's probably because even though the citrate can't go anywhere, or can't go anywhere in the citric acid cycle, it can be exported to the mitochondria, to the cytoplasm, where it can give rise to fatty acid synthesis. And cytoplasmic acetyl-CoA comes from citrate, and it goes to fatty acid. It also goes to the synthesis of acetylcholine, among many, many other compounds with acetyl-CoA. And the reason that's interesting is that one of the chronic problems in Barth syndrome is diarrhea. Any of the parents will tell you here that is a major problem. And we saw a child in 2010 in our little consultations who has post-transplant was having a terrible time with diarrhea and volume control and was having trouble maintaining stable levels of the chemotherapy drug. I forget which one it was. So I said, well, maybe your child is overproducing acetylcholine, which is the primary neurotransmitter of the gut. So I advised the parents to take all the citrate, all the extra citrate out of the diet. And sure enough, the diarrhea stopped. And the blood level of the chemotherapeutic agent was stable from there on out. Conversely, there are mitochondrial diseases where we see low citrate. The block is further up. And those patients have pseudoobstruction. They have decreased motility. And if we give them citrate, it restores motility. So the citrate level, a disturbance in the citrate level, depending on the type of mitochondrial disease, can lead to excessive gut motility or decreased gut motility. And pseudoobstruction in mitochondrial disease is a very serious problem. It can be lethal. And the first time I treated a child who had pseudoobstruction with citrate, she went from 1 stool every other day to 10 stools after I gave her the polycitra. So I cut back on the dose a bit. And she's been fine ever since. And I haven't used it that many times, but I've used it enough times to see that it usually works in a mitochondrial problem. I'm not saying it should be used for constipation in the average child, but it was very striking. So understanding, keeping this in your mind, which I hope may come a little bit out of this talk, one can think a lot about the consequences of mitochondrial dysfunction, the cellular consequences of mitochondrial dysfunction. So this is what we also measured at the 2008 conference. Because lipids are involved, and there is some thinking that there might be some function of TAZ in other membranes. And I don't really know what the status of that thinking is, but we went ahead and looked at fatty acid in red cell membrane. So this is a standard clinical test looking at mostly for looking at DHA levels. But the red cell being a long-lived cell, the fatty acid composition, that is the fatty acids in the phospholipids and triglycerides in the red cell membrane, generally reflect the body composition of fatty acids in phospholipid. And so what we find here is, this is Z-scores, standard deviations, clearly increased total saturated fatty acids are increased, and some of the polyunsaturated fatty acids, like DHA, are decreased, and the total N-6 and N-3 are also decreased. This is particularly interesting because the fatty acid, endogenously made fatty acid, is saturated fat. And so that the accumulation of saturated fat throughout the body may be secondary to this block and accumulation of citrate from the citric acid cycle. And it's interesting to think that the sort of the pudgy cheeks that babies, that boys with Barth syndrome have, we in the NICU, they were called preemie cheeks, and you saw that in growing babies who were on, this goes back many years, on MCT oil. And MCT oil is saturated fat, which can be elongated into longer chains, also metabolized. But what I'm saying is that this obvious abnormal deposition of fat that we see in Barth syndrome may be related to the diversion of citrate into fatty acid synthesis. One of the things that we do from a dietary standpoint, knowing that most fat is used to supply energy to muscle, and that muscle mass is decreased in Barth syndrome significantly, and type 1 muscle in particular is basically not working or hardly working at all. So the need for fatty acid is reduced, and therefore a child with Barth syndrome should be on a relatively low-fat diet as a routine. And I'll show some other things. And obviously we have trials going on now and treating with arginine 2 to correct heart function. And the short story about that is that it works. It's not quite as easy as you might think to get the arginine level stable and up to normal, but if we can achieve a normal arginine level, the prealbumin level goes up, showing normal protein synthesis, and the heart function normalizes. So what else can we learn from this fatty acid abnormality? So the fatty acid distortion here. So this affects membrane structure, and receptors in the cell membrane are sensitive to fatty acid composition. A lot of the research on so-called metabolic syndrome in adults is on decreased insulin response and the fact that changes in the Western diet may modify hormone responsiveness because of the change in the phospholipid composition of the cell membrane. And if one looks at the literature, the in vitro literature or animal literature, where they manipulate the diet and change the fatty acid composition, then you do see that high saturated fat for a number of hormones tends to decrease hormone responsiveness. So in terms of so that got me thinking about neutropenia. So neutropenia in Barth syndrome is very unusual as well. And it is, one can show abnormalities in mitochondria in the bone marrow, and there's a lot of speculation about apoptosis and that the cells are abnormal and are dying as a result. However, when you give GCSF, or when a child gets an infection and is making their own GCSF, the white count goes to 20, 30,000 with no trouble whatsoever within 24 hours, and the white cells are perfectly functional. And I can tell you from someone who tries to culture abnormal cells in the laboratory, the fastest way to make cells die is to make them grow. So if you give GCSF and make the bone marrow grow, and if the neutropenia were due to cell death, you would not get this exuberant leukocytosis that one sees. So it suggests that it might be an abnormality of hormone response, that the responsiveness to GCSF might be the primary problem. So we also, of course, collected CBCs at the time of the meeting. So I did a simple correlation of the white count with the fatty acid composition. And this is not the neutrophil count, not the ANC, it's what I call the neutrophil index. And it's calculated by subtracting the monocyte count from the neutrophil count. And I can explain it, I can hardly explain it to myself each time, but there's a logic behind that. It's basically the fact that the monocytes go up when the neutrophil count goes down in the average Barth patient who cycles. But if you look at the variation between patients, the total white count will be different. So if the white count is at a high point, the monocyte count will be at a low point. So if you subtract the monocyte count from the ANC, it gives you, it corrects for some of the deviation between patients. As I say, it may seem crazy, but there is a good logic for doing it. And if anyone wants to have me go through it. But anyway, what you see is, so this is the ANC here, and this is the linoleic acid concentration as a Z-score. And linoleic acid, which was tried as a treatment for Barth syndrome, makes the white count go down. So as the linoleic acid concentration goes up, the white count goes down. And we also saw, looking at methylglutaconic acid, that as the linoleic acid concentration goes up, the fasting level of linoleic acid in blood, which also reflects body linoleic acid, as linoleic acid goes up, methylglutaconic acid goes up. So the biochemical severity increases with linoleic acid. Oleic acid, however, it looks as if that helps the white count. total omega-6 will give you the same correlation. Oleic acid gives you the opposite, that as oleic acid goes up, there appears to be a correlation with that, and again, total omega-9. So this is an argument that the problem in the neutropenia is a hormone response problem, that it's a membrane abnormality that decreases the response to GCSF. So then we have to think about growth. The growth in Barth syndrome is, as I've said before, very unusual. And the boys are very small. By the second year, they're usually well below the third centile, and they grow at variable rates. This is a child who went into heart failure right here, and then the heart failure was corrected, but he still stayed below the third centile. But look at this boy here who stayed well below the third percentile, had a late pubertal growth spurt, and he reached a normal adult height. And I'm sure you've all seen the tall Barth boys around here. There's no shortage of them now. And this shows others who went through puberty. One of the problems here is that, in effect, what happens at puberty, they just start growing both for puberty and catch-up. So the growth velocity in a child with Barth syndrome during puberty is twice the growth velocity during puberty in a child without Barth syndrome. And as you can imagine, if we're dealing with a nutritional mitochondrial disease, it takes a terrible toll on the boys, and that's one of the times that they go into heart failure, is during puberty. And it probably is nutritional in the sense that they are not keeping up with their increased protein, their increased arginine need. I forgot to mention that one of the things that was found at the 2008 meeting in taking dietary histories, that many boys self-select a diet that has between 3 and 5 grams per kilo per day of protein, which pediatricians here will know is huge. And they're basically, you could argue, they're eating enough protein to get their arginine level up to a minimal survivable level. But it doesn't help them. They end up eating a lot of protein, of amino acids that aren't needed, so they get fat. So these little boys running around that you see who are very chubby, not only may they be making more fat of their own, but they are eating a lot of calories just to keep their arginine level at a normal point. There's one mother who's an engineer, and she engineered the diet of her boy with Barth syndrome so that it had all the arginine, so that he had a normal arginine level by eating arginine-rich foods. And his growth is normal, and his heart function is completely normal, and has been so for 3 years now. So it really is a nutritional problem. But the question is, to get back to the real question, why does this happen? Well, the other thing is some of the boys here who have growth delay have been seen by endocrinologists, and they have classical constitutional short stature, which means their growth, their bone maturation is appropriate for their height. So if they're at the height age of, if you're here at a 10-year-old with a height age of a 6-year-old, the bone age will be 6 years. That means that they have the full potential for growth, and that's a classic setting. Most often pediatricians say, well, you'll grow up eventually. But some of the boys have been given growth hormone, and in most cases it doesn't work. And that's very unusual. In constitutional short stature, if you give growth hormone, they grow. And that is, again, it says, is this a hormone response problem? And that's what I was saying earlier, that the growth delay is out of proportion to the metabolic disease. So at this meeting, we're collecting more endocrine data to try to understand what to try to prove this hypothesis that it's a growth hormone response problem. Why does they grow so enormously at puberty? Well, what happens at puberty? Testosterone is produced, and what does testosterone do? It increases the sensitivity to growth hormone, or to the growth hormone. It increases the responsiveness of the growth hormone receptor system. So they reach puberty, testosterone goes up, and this relative block in growth hormone response is relieved, and they just take off. And so we're trying to establish that. Our goal, of course, if we can regulate the fatty acid composition of the red cell membrane and show that that really is the problem. Of course, we're getting data on growth hormone and growth parameters and going to correlate that with the fatty acid composition of the red cell membrane. If we get a correlation with that, then we have an argument that if we could try to engineer the diet to change the fatty acid composition, we might change hormone responsiveness. So our conclusions about these kind of studies in Barth syndrome are cardiomyopathy, I believe, is nutritional due to endogenous amino acid depletion. As I said, the major effect of cardiolipin deficiency is impaired import of citric acid cycling enzymes. The TAZ deficiency affects non-mitochondrial lipids. As I showed, it may be from the oversynthesis of saturated fat. And the neutropenia and growth abnormalities could be caused by abnormal receptor response. What I want to turn to briefly now is to say how enormously figuring this out in Barth syndrome has changed the practice of treating mitochondrial disease other than Barth syndrome, real mitochondrial disease, and in ways that can be life-saving. And it's really been, I've been frankly exhausted running around retesting all my patients and figuring out what they are deficient in and treating them. And it's quite remarkable how the Barth mitochondrial paradigm applies to other mitochondrial diseases, which I'll show here. One, I just want to mention also that and this will come to the mitochondrial treatment, in regressive autism, these are children who are perfectly normal up to a year of age. They get an infection, and they, over a relatively short period of time, lose their language and social skills. Classical regressive autism. As you all know, there's quite an increase in autism, and it is in particular regressive autism. Well, we analyzed the amino acids in the same controlled way in autism, looking at regressive versus non-regressive autism. And again, here are our normal ranges for 4-hour fasting. And there's no question that alanine, which is a marker for pyruvate, and glycine are increased. That combination is a biochemical marker for complex 1 deficiency. So we looked at citric acid cycle intermediates in one of the patients, and we can treat this kind of autism. If autism is due to a mitochondrial problem and you get to them early enough, you can treat the mitochondrial abnormality and they get better. And the primary block, the major block we see is at this level here, but there's also some increase in alpha-ketoglutarate. But we treat to augment complex 1 activity. And complex 1 is required for both alpha-ketoglutarate dehydrogenase and isocitrate dehydrogenase. So that's we sometimes see only isocitrate increased, and sometimes we see both increased, and often with the aconitate. But the amino acid profile and this TCA profile suggests a block in complex 1. We treat the complex 1 deficiency. The behavior of the children can change overnight sometimes, and as you can see here, with treatment, the levels came down. It's not entirely normal, but the child is now developing fairly normally, and the real abnormalities in the TCA cycle improve with treatment. Multiple sclerosis if you read the literature now, you see that there's increase in suspicion that mitochondrial dysfunction may play a role in multiple sclerosis. And we have been collaborating with one of the doctors at Hopkins. And indeed, this just shows appears to be a more specific block at alpha-ketoglutarate dehydrogenase. And when you look at a larger group of patients with multiple sclerosis, you can see that quite a few appear to have an abnormality there at alpha-ketoglutarate. That's important because if there's a block here at alpha-ketoglutarate, this large source of arginine is not available. From a total amount available in the body is a lot more arginine than methionine. So methionine, isoleucine, and valine are then, at least potentially, can supply succinyl-CoA to keep the citric acid cycle going. And and I'll come to later in dealing with another syndrome with a block at the same point, what the significance of that and methionine is. We're just beginning our studies on MS. We also see a number of syndromes that clearly do not have a primary mitochondrial problem, but because the children deteriorate in some neurological way, we have to suspect there may be some mitochondrial dysfunction. And to make a long story short, there are very few ways for brain cells to die without damaging the mitochondria at some point in the process. So we looked at a boy with Pierpont syndrome, which is probably a chromatin regulation defect. The gene hasn't been found yet, but it probably is a chromatin regulation abnormality. And the boy I follow, there are only like 6 cases in the world. The boy I follow had an abnormal amino acid profile. It looked like complex 1 deficiency, and indeed he has a block at isocitrate dehydrogenase. I treated him in the same way, and he started walking for the first time after a few months, and it's just his physical therapist and that can't believe what he's doing now. So it really treated the underlying mitochondrial dysfunction. Exactly why it's abnormal, we don't know. I say this is a chromatin regulation problem, which another syndrome that is an abnormal chromatin gene regulation is Rett syndrome. And we also find mitochondrial abnormalities in Rett syndrome. And that's actually been published for many years, but it's never been fully understood. Adrenoleukodystrophy turns out to be a systemic mitochondrial disease. This is the Lorenzo-Zoil disease, where they have abnormalities in very long-chain fatty acids. And I had studied a long time ago Zellweger syndrome, which has a lot of the same biochemical abnormalities. And I studied mitochondrial function in them, and I found that there was a block at this level here by oxygen consumption studies. And I've not worked with adrenoleukodystrophy, but Hugo Moser, who passed away recently, is at the Kennedy Institute, of course, world famous for this disease. And what it appears to be is the very long chain fatty acid that accumulates in adrenoleukodystrophy causes a block at this level. And methionine is involved. And again, I'll show you an example of how I think methionine is involved in this. These are boys who are asymptomatic, but are from a family with classical X-linked ALD. And almost all the boys have this abnormality. Smith-Lemli-Opitz syndrome, we see also abnormalities and enormous response to treatment. The problem here is that there's accumulation of a fatty acid in Smith-Lemli-Opitz syndrome that it causes free radical generation. And in these children, I treat them with an antioxidant cocktail. And again, I won't go into detail of the number of clinical changes that occur, but one of the problems in this condition is a problem in making cholesterol. I mean, the total cholesterol level is low. And when we treat with the antioxidant cocktail, in about 6 months, the cholesterol level is normal, and the child is putting on muscle mass. And again, some of the children have walked for the first time after being treated. So no one would have thought that there was a mitochondrial problem, but our having set up citric acid cycle intermediates as a way of understanding Barth syndrome have given us a tool to understand all of our patients with mitochondrial disease. Now, I don't expect to go through this in detail, but this is a very important study which has gotten I've never seen anyone refer to this study, but it's absolutely crucial to understand what this group did. It's from 2007, and they took Leigh disease cells. Leigh disease is a severe mitochondrial disease, brain injury. Most of the children die at a fairly young age. And they did what's called isotopomer analysis, where they label substrates, And they can trace individual carbon atoms all the way through the citric acid cycle. And they measured all the fluxes. So this is the flux in, this is the flux out, and that's the flux in for each of these compounds. So they basically measured all the citric acid cycle intermediates, amino acids. It was really a phenomenal study. And they concluded, what they concluded was the levels of the citric acid cycle intermediates, as you can see here, stay normal. And again, that's the same theme, that ATP synthesis stays normal in this situation. But what happens is this measures substrate uptake by mitochondria and substrate secretion by mitochondria. And look what's really big, arginine and methionine. In Barth syndrome, there is evidence of methionine uptake, excess methionine uptake. Arginine stands out more. But in a mitochondrial disease where the block of that alpha-ketoglutarate, methionine is going to be it may be depleted. Well, what is methionine? Then you ask the question, what does methionine do? Well, methionine is, of course, a methylation amino acid, and it's involved in all sorts of methylation problems, and it's tied intimately into folate metabolism. And I just show here that in most Lees disease cells, we see a block at that point in alpha-ketoglutarate, which is why the methionine, I believe, was, again, to remind you that methionine is after alpha-ketoglutarate comes in there. These are also, you see that graph, these are also consumed, but especially methionine. It's a very minor component compared to many of the other amino acids as a total fraction of body amino acids, but it seems to be picked on, so to speak. So it's involved in folate metabolism, and one of the signs of folate deficiency is a large red cell size, so-called macrocytosis or megaloblastic anemia. So I looked at the red cell size in some of the patients with Lees, and sure enough, there is a clearly abnormal progression in the size of the red cell, as if there's an increasing deficit of methylation is one explanation. Folate levels are very high in the patients. They're not low. B12 levels are normal, and this probably is what's called the folate trap, and that complex 1 in particular, but mitochondrial function is involved in the recycling of methionine and folate, and this may reflect a progressive folate trap. So is methionine necessary? So one way of restoring methylation rapidly is to give folinic acid. It's active folate. It doesn't have to go through the cycle to remethylate the folate. And here's one boy with Lees disease who did quite well. I don't know what his CBC was, what his MCV was back here. He started treatment back here, and this is the first blood count that I have on him. I suspect he might have been higher, and he got better in this period here. But then he started the plateau in his treatment. I've been able to treat Lees disease long-term and long-term survival using standard mitochondrial cocktails, but he didn't respond in the usual way. He got better at first, but then he started getting worse, and his MCV was climbing. I gave him folinic acid, and the MCV dropped immediately, and he was having what are called central hypothermia, central fevers, which is seen in some in brain injury. And when we started the folinic acid, the fever stopped that day, and he since has continued to improve. Now we know that we've treated other Lees disease patients with methionine and gotten the same effect. And if you again collect the amino acids in a very careful manner, the methionine level is mildly decreased. It's not as decreased as you would think, because we also see that the conversion of methionine to cysteine, which is a normal process, is blocked in these patients. And that's probably because methionine is deficient and the body is doing everything it can to conserve methionine. So the studies in Barth syndrome, and especially the study of the Barth syndrome cells, of the Leigh disease cells, has told us that in mitochondrial diseases, that ATP deficiency may not be the primary cause of cellular damage. That in Barth syndrome and other classical mitochondrial diseases, it's extra mitochondrial substrate depletion that is doing the harm. I'm not saying this is universal, but ATP synthesis is preserved by any means that can be done. We'll have to remember that mitochondria are intracellular parasites, so they're going to do a number on the rest of the cells to keep themselves going, and that's what we're finding. And again, to emphasize that simply doing careful plasma amino acid analysis, measuring TCA intermediates, gives you immediate insight into what's going on in that mitochondrial disease. Most of my patients, I have no idea what their molecular defect is. I don't need to. I look at the biochemistry, tells me how to treat it, and I treat it, and then at my leisure I can figure out what the molecular defect is. So I wish to thank all the Barth syndrome families here for not only getting me interested in Barth syndrome, but also in helping all my other mitochondrial disease patients. Thank you very much. I think it's with five minutes for questions, right?
Okay.
Questions?
Questions for Richard at this point?
Maybe 4 minutes for questions.
Fascinating study. I asked a related question about mitochondrial deterioration in aging. Have you analyzed the amino acid profile in aged patient, because the tetralinolenyl cardiolipin level also go down as we get older? Whether the similar defect also exists, like depletion of arginine. And another related question is about people who are obese and diabetic have elevated level of leucine. So that's another related issue, whether you have any answer to those defects.
As I say, I'm a simple clinician, and I do amino acids and I do TCA intermediates, and any real research is done by others, I hope. So they're very good questions, of course, and there definitely are mechanisms by which cardiolipin is depleted in other systems. And I guess what you're saying is correct, that maybe one should think of, by doing these kind of studies, you in a condition where you think there is cardiolipin depletion, maybe you'll find the same abnormalities and then end up with a treatment. There are many mitochondrial diseases that I've found respond to methionine. They need the rest of the mitochondrial treatment too, but giving them methionine makes a huge difference. One of the things that we see, for example, is excess weight, excess fat accumulation occurs. They become obese even though they're not eating a lot of calories, and it's largely because they can't metabolize fat. When we start them on methionine, the fat just melts away. It's just amazing. Any other questions?
Richard, if the ATP synthesis is preserved, which you found and our people found too, then that of course requires that the TCA cycle is largely intact.
Yes.
This is sort of an internal contradiction in your hypothesis. But I guess what you're trying to say is that there might be some sort of moderate abnormalities in the TCA cycle that we need to figure out. And related to that, my question is, how well do the plasma levels of citric acid cycle intermediates correlate with the actual matrix concentrations?
With the actual what?
Matrix concentrations.
The actual what's going on inside the mitochondria?
Right.
Well, those are the questions for scientists to ask.
That's why I'm asking that.
Right. So I mean, that's the question for scientists to answer. I'm looking at this from what is making my patients sick, and this could be completely wrong, but at least I can treat my patients now.
I think there is one relatively straightforward way to find out whether or not it is completely wrong, and that is to take mitochondria that from any cell that has Barth syndrome, we can go to yeast or to any other system, I guess, then see and study the citric acid cycle by standard tracer kinetics. And the two questions would be, is there a change in the steady state concentration? And second, is there a change in the flux through the cycle? These, there are standard methods for this. Yeah. They go back to the 1950s, actually, when these things were elucidated.
So do I. I think Dr. Greenberg is the expert in that area, and she's definitely working on that approach. And we've obviously been in communication about some of these findings, and she's very interested in looking at this aspect of mitochondrial function.
I'm definitely not an expert in this area, but this is an area that I very much hope that we're going to become expert in. And this is, in fact, related to the experiments that my student is hopefully going to be doing in your lab, Michael. So,
and I will answer
this. You will answer the question. So, and actually, I'll be talking about that in later this afternoon.
Okay, great.
Have your program.
Hey, a very quick question about the potential origins of neutropenia in Barth syndrome patients.
I'm sorry, I was...
Sorry.
Again?
Yeah, I was just wondering about the origins of neutropenia in Barth syndrome patients. Like you said, it's very unusual in terms of being able to respond to GCSF pretty easily, as opposed to some other bone marrow failure syndromes where patients can get all GCSF we want and they will never respond. So there is a potential for the bone marrow to increase the synthesis of neutrophils, either in response to GCSF or response to infection. And I was just wondering if we know a little bit more about the origins of that and why those patients become neutropenic. Is it, do you think it's the cell cycle problem? Do you think it's the problem with release of neutrophils from the bone marrow? Or do you have any good idea about what could be the molecular background for that?
Yeah, I'm sorry, I had trouble hearing some of that.
The rationale for the neutropenia.
Yeah.
An explanation for...
Again, just to repeat that my hypothesis that we're trying to sustain with the laboratory studies this year is that it's an abnormality of GCSF hormone, GCSF response, that the receptor, GCSF receptor, is hyporesponsive because of the membrane, the abnormal membrane that it sits in. There's a very strong precedent for this in adrenoleukodystrophy, where there's accumulation of very long-chain fatty acids. And one of the common presentations of adrenoleukodystrophy is Addison's disease. And that's a failure of ACTH to stimulate adrenal function, low levels of cortisol. And it's been shown that the cause of that is that the accumulation of very long-chain fatty acids in the adrenal cell membrane reduces the receptor response to ACTH. So there's a very good precedent for saturated fatty acids, very long-chain saturated fatty acids depressing receptor response. So it's partly the peculiarities of the neutropenia in Barth syndrome and this precedent for receptor response regulation. Do you want to move on to Colin?
Move on to Colin.
Okay.
So there could be a way to test this, actually. If you think, for example, if you took Barth syndrome lymphoblasts and incubated them with different types of fatty acids, and then hit them with colony-stimulating factor and see whether they go into apoptosis any faster or not. So that might be a simple experiment to at least put you in the right direction for the types of fatty acids that are involved as well. And if the receptor issue is involved as well.
These are, I'm sure these things are testable by people who have the resources for doing it. And I hope that I will at least stimulate some interest in trying to prove me right or prove me wrong. One way or the other, it's going to help Barth syndrome. Okay? Yeah. You want me to introduce? Yeah, I'm doing it, sure. Oh, yeah, I don't need that.